Hi, I'm Mr. Wilson. Welcome to worksheet number 2, Explained. I hope you had fun doing worksheet number 1 and you are ready for some more useful content. Today, I'll be explaining some questions from the topic cell, as is covered by the CSEC syllabus. I hope you liked, share, and subscribe as we work to build this community of A-class students. Grab a friend and let's go. So today, as said earlier, we are going to be looking at cells, worksheet number 2, and those persons who are just starting out in HSB, Human and Social Biology, could join along in doing this worksheet. I'll be explaining the question as would have been expected by the exam. So we're going to be looking at some keywords that are color-coded. So it is CSEC Biology with Mr. Wilson, Life and Environment Explained. Alright, one word we're going to be looking at today in our question is compare. When we compare, what do we do? Of course, we look at similarities and differences. So to compare is to look at similarities and differences. Now we could compare a male and a female. Of course, they have 2 hands, 2 feet, that type of a thing. Now what are the differences? Of course, you could name many. Now, this question is particularly important to me as it outlines the use of a table and what is expected when working with a table. The question reads, using diagram, you're supposed to compare the structure of a generalized plant cell, animal cell, and microbe from those named in your biology syllabus. The following organelles are to be included. Now, please observe drawing rules as much as possible when you are going to be drawing. Now, what is expected of this question? Let's find out. You are going to be drawing all 3 cells. The generalized plant cell, the generalized animal cell, and that cell that would have fit the description or the organelles as is outlined under microbe. You're going to be drawing all 3. But we are interested today in the structure, the makeup, the structure. We're not really looking at the function as it were here. We are looking at the structure. So you're going to be drawing all 3 and then you are going to be labeling. In this case, I just want you to label. There is no need for annotation. We are going to be taking it a step at a time. So today we are just going to label. So you're going to be following the rules for labeling a diagram. Now, this is a diagram and not a drawing. So for this bit, magnification is not needed. However, it is very important that you ensure that the lines that you use to label they are parallel to the top of the page. You are using lowercase letters only to label. The lines are touching the organelle that you intend to label. The words are correctly spelled. And your diagram has a title that is descriptive. Alright? Please be reminded you want to follow the drawing rules as much as possible. If you look below in the description, you will find the rules for drawing. We have a video out on drawing rules. You want to go and look at that if you really don't remember what the rules are. Let us look at this table. Now, one thing we usually remember for CSEC is that a table is bounded by 4 sides. Tables usually have a title that is descriptive and it's usually written in uppercase and underlined. Now, if you look at this table here, we have 3 columns. And for each column, the column has a title. So you'll observe we have plant cell, animal cell, and the microbe. And then we have the listing of organelles that we'd want you to use to label the cells. I want to point out something. Look at the diagram. I'm using my mouse. If you observe this cell here is empty. Nothing is written in it. Now the convention of using a table is that once there is an empty cell, you need to put a little dash, a hyphen in this blank cell so as to indicate to persons who will be reading your table that nothing was left out, nothing was deleted. This is just a placeholder. So if you observe, we have 1, 2, 3 blank cells, and these cells have the little hyphen in these cells as placeholder, so you can't interpret or misinterpret or go writing anything there. It just tells you that this cell should not have any value or character. I do hope you understand that bit. So you want to draw your table, and the table should not be touching the margin in your lab books. Please do not draw a table to touch the margin in your lab books. You are going to be using a ruler to draw your table. You are going to ensure that your table, it has a title, and your columns and rows where needed, they too must have header title and what we call header title, and we might have header rows. I do hope that that is clear. There's something else I want to point out which is not in this table, but if you're going to be using values, like for example we're using degrees Celsius, all you need to do is to write the degree Celsius just in the header column here. It should not be repeated in the cells. All that is repeated in the cell is actually the value. So if we are looking at 78 degrees Celsius, the temperature at which alcohol would boil, you would have 78 in the cell here, have 78 in whichever cell here, and the degree Celsius would be right here in a bracket. Do hope we understand that bit. Let's move on. Distinguish. To distinguish is to identify differences. To distinguish is to identify what is it that make this thing different. We're looking at difference now. All right, so let's hop on to this question and see how well we could handle this one. We want to distinguish between the cell wall and the cell membrane. Remember, in the description below, you're going to be finding the link to the lesson on cells, and you can go there and watch it how many ever time you so desire, so as to ensure that you score full marks. Now I want you to observe this question carefully. If you observe, we have used the word while, because at no time should we try to do a distinction and it ends up being just two definitions. Where you write two definitions, what you've answered to is to define two words. So you don't want to just define two words. If you are going to be defining, you are going to define, and then you are going to use the word while, which makes the comparison or makes the distinction there. X is that, while Y is that. Please make sure that you go through these again. We have explained these before in worksheet number 1. We want you to go back through the worksheet. We want you to grow gradually. We are going to be going back to the front of the syllabus, but we thought that moving off with cells and characteristic of living things were a very good topic just to get you in the mood. With COVID and all that, you need a little rest from the hard work, so we are going to take our time, and then we gradually and let things get a little more challenging. But for now, let's enjoy what's in front of us. So we want to also distinguish between the mitochondrion and the chloroplast. Remember, in our last lesson we were trying to figure out about mitochondria and mitochondrion. Those words, we were trying to work around them, what they were. I do hope that you have done your homework, because you might be seeing these words in a very uncomfortable area, and you wouldn't want to know that you don't know the difference. Now I want you to watch the video in the description. This is going to be video link number 3. I want you to watch the video on the microscope in the description below, and I want you to name 4 parts of the microscope. Any 4 parts you so desire to name. But I do encourage you not only to watch the video to identify the 4 first parts, but to ensure that you have a good grasp, a good understanding of all the parts of the microscope, and of course how to use the microscope. You can't be a biologist that can't use a microscope. Come on guys, come on guys. So we're going to be watching that video right to the very end. I think we're doing well so far. This is going to be a very short lesson, a very short research for you, for those persons who will have to do a research, and for those persons who have done the worksheet already, I can assure you within a matter of minutes you will be all over and done with this worksheet. So we want you to name. In our last worksheet we said name. If I were supposed to ask you, what is your name? All right, what is the name of your school? It wouldn't be any epistle, any long thing. You just give me the name of your school. In the last lesson we said that my school was the cover page I. That is all that it is. Just give the name. Next question. We're now asked to state. What does state mean? When you state, you are providing factual information that is appropriate in length. Come on guys, we are doing CSEC. We're not writing for a PhD paper, so we are not going to be writing everything there is to write about this particular topic. We are going to ensure that our answer is within range of the marks. Now, the only way you are going to be able to do that is if you have good grasp of the content. And to have good grasp of the content, if I am allowed to go to the Bible, it says study to show thyself approved. I don't know if you're a Christian, probably you are not, but if you study and practice, you get better anytime at all at what you do. Of course, you should get better once you're practicing. So here we want to state the function of any two parts of a microscope. See, you have watched the video, you named 4 parts earlier. Now you're going to tell us the function of any two parts of a microscope. Now the marks on this worksheet, your teacher might choose to adjust the mark. They might use a different mark scheme. I don't know. These are just pretty much suggestions. So before you do or while you do, you want to consult with your teacher to ensure that you understand the rubric, you understand the mark scheme, that you're scoring full marks. Let's move on. We want to relate. Now this is the big one. Relate. When you're relating to something, you are showing the relationship, the connection. What connection there is. Let's look at this question. We want to relate the structure of a following organelle to their function. If you looked somewhat at question number 1, we spoke about structure, the makeup. So now we are relating structure to function. And let us use some analogy I hope everyone will know. Look at the design of a boat. I'm assuming everybody would have seen a boat, whether a real boat or one on television, but you would have seen a boat. Now the design of the boat, the structural makeup of the boat, it suits it for its function in comparison to a motor car. A motor car has 4 wheels for most cases, and it is suited for its function. That structural makeup, that design, it allows the car to drive on the road. My question to you, would the boat be able to sail on the road where there is no water? Of course not. Would the car be able to drive across the sea without any modification? Of course not. So these are examples of structure, the relation of structure to function. So the car is designed with these 4 tires so as to enable its function. The function of a motor car is to drive on the road from point A to point B, taking persons or cargo from one place to another. The purpose of the boat, on the other hand, is to sail on water, not to drive on the road. So here we have structure, the way it is made, the way it is arranged, versus what it does, the function. So now we're going to be looking at this question, and we're going to be looking at the chloroplast. What makes the chloroplast so suitable for its function? What about its makeup? What makes the mitochondria, remember when we looked at these, what makes the mitochondria suitable for its function? You might have to go a little out of what we presented, but make sure when you're answering, you're comfortable enough to say that because of its structure, it is able to carry out this function. The vacuole is pretty much the same. You are going to look at the vacuole, its makeup, its structure, what it is like, and then you are going to say how this has made the vacuole ideal for its function. And the same is true about the nucleus. Now we could say that the nucleus carries genetic material. It has chromosome, that type of thing. You could go around and look at the makeup. What is it like? Now, if you have ever read or you've ever gone through, for example, the red blood cell, where we say it's like a biconcave this, and that depression there, we talk about oxygen moving, you understand structure and function. Are you with me? So let us look at a spoon versus a fork. The design of the spoon, the structure, it allows you to take up liquid. But would you say the same is true for a fork? No. So here we have it, structure versus function. So now you're going to be you're supposed to relate the structure to the structure of the following organelles to their functions. Do we understand? I hope we do. So this is not a question that you are going to just write out the function. You want to tell us the structure is like that, or the structure is this. As a result, it is able to do that. All right? Good. Perfect. We are doing well. And if you have any challenges, you could consult with your teacher, or you could just leave us something in the comment and we will provide further explanation in videos to come. Differentiate. Another big word. You are now going to tell the difference. To differentiate, you are going to tell the difference. I'm hearing somebody saying, why all these words in yellow or gold writing, and sir keep emphasizing these words? These are likely to be the words that are used to coin the questions for your external exams. As such, you have to make sure that you are comfortable with these words and you understand without any shadow of a doubt what these words mean and what is expected when these words are used. So let's look at a question. You are supposed to differentiate between plants and animal cell. In so doing, relate the structure to function. So we are going to be looking at the differences between a plant and an animal cell. But not just the difference, we want to look at the structural function. The structure, how does the structure allows it to be what it is and to carry out whatever role it carries out. Alright, so if you look at a plant cell, how rigid it is versus you look at a animal cell. Alright, and if you go in osmosis, you would have a better appreciation when I speak about cells and how cells are rigid because they have the cell wall versus the cell membrane. Now I want you to do some discovery. So I want you to look at the structure of the animal cell and its organelles, and I want you to look at the function of these organelles. So you are going to look at the makeup of these organelles and you are going to be looking at the function. And having done your reading, you should be able to say, because organelle has this structural design, it is able to do that. You might be familiar to different types of shoes. There are some that would have a lace, a shoelace, we call it shoelace, and there are those without lace. Because of the structural makeup of the shoes with lace, you are able to tighten the shoes with the lace, or probably you want it to be a little slacker than it is. So the structural makeup will make a big difference in how anything functions. Now, this is another very interesting question. We want you to use the following to construct a flow diagram to show hierarchy. Now, here we have organisms, ecosystem, tissue, cells, community, that type of thing. So we want a flow diagram that is going to put these things in order. What comes first, what comes second, so on and so forth. So for example, if we were counting to 3, we'll start at 1, then we get to 2, then we get to 3. So here we have these things all jumbled, and you are going to put them together to say what comes first, what comes second, then, and that type of thing. And we are going right up to a ecosystem. I do hope that this lesson was very beneficial to you. I do hope that you have learned. In our next lesson, we are going to be covering a very short lesson, cell specialization in multicellular organisms. We're trying as much as possible to make these lessons very short so that you have enough time to watch them with the limited credit that we're hearing about, or the limited service, or the attention span. We are looking at all these while we are creating these training videos. Now remember to like, share, subscribe, and leave a comment. Now hit the notification bell so you will be notified when we release a new video. Until next time, keep studying. Later.